Hi, and welcome to Chapter 14. In this chapter, So how did we discover DNA and eventually figure out that it is our genetic material? It really starts back in the 1800s with this doctor, this physician, Friedrich Miescher, who was a curious guy. He decided to look at pus from hospital bandages because he knew that this pus was high in leukocytes, these white blood cells. And what he did was he looked inside the nuclei of these white blood cells and he found phosphate-rich acidic compounds that he named nuclein because he isolated it from these white blood cell nuclei. Eventually, this is now known as DNA, or deoxyribonucleic acid. About half a century later, we have bacteriologist Frederick Griffith, who reported the first experiment on something known as bacterial transformation. He discovered that something is transferred from cell to cell, or you could say from the environment into the cell, that provides the cell with instructions to build new cellular components. And we didn't know what it was yet. We did not know it was DNA, but this was one of the first major experiments to show that information can enter the cell and allow it to build new structures. So Frederick Griffith performed his famous experiment using bacteria named Streptococcus pneumoniae. So this is a type of bacterium that causes pneumonia. Interestingly, there are two types of Streptococcus pneumoniae. There is one called the R strain, R meaning rough, and there's one called the S strain, where S stands for smooth. And rough and smooth really refers to how the streptococci bacteria appear when they're growing on a petri dish. So the rough strain happens to be rough because it does not have something called a capsule. There's no capsule. If it is injected into something like a healthy mouse, it will not cause disease. The mouse will remain healthy because it is able to fight off these bacteria, these foreign cells, and nothing happens to the mice. But the smooth strain is pathogenic. It's disease causing. And the reason for this is because these bacteria do have a capsule, which is a sticky layer outside of the cells that makes it appear smooth, but also allows it to stick to our body tissues and hide from our immune system. So when he injected living S cells, the pathogenic or disease-causing cells, into mice, then the mice die. What he did next was take heat-killed smooth cells. And I know that when I heat these cells, it'll kill these bacteria. And he injected it into mice, and the mice stay alive. They're healthy. And that kind of makes sense, right? I destroyed the disease-causing bacteria before I injected them into the mouse, so the mouse survives. Finally, he did something that was pretty interesting, where he took the heat-killed S cells, and he mixed it with living R cells. So separately, they both had no effect separately. But when you mix them together and inject them into the mice get sick and die. And what's even crazier is when he examined the blood of the mice, he found living S cells, which is really weird because he did not inject any living S cells. He only injected living R cells and dead S. So how did you get live S cells afterwards? He concluded that something was released by the heat-killed S cells. So let's say this is an S cell, and you're killing it. And maybe something is released, so some kind of particle is released by those dead S cells and enters the R cell, allowing them or giving them the instructions to produce some kind of capsule, produce that capsule that turns them into S cells. And this is known as transformation, when bacteria pick up this genetic material that we did not know as DNA yet and become transformed. They can take those instructions and build new structures. The next big experiment that led the way to the discovery of DNA was performed by Avery, MacLeod, and McCarty. So they wanted to know what that transforming particle was that was discovered by Frederick Griffith. So what they did was they took those S cells, the Streptococcus pneumoniae, the disease-causing one, pathogenic one, and they exposed these S cells to different enzymes. And these enzymes' purpose were to destroy different macromolecules found within these cells. So, for example, for one set of the S cells, they added proteinase to get rid of all of the protein, and then they added in live R cells to see if transformation occurs. So what happens at the end is they do find living S cells. So something that is not protein is entering the R cells to turn them into live S cells. So protein is not the transforming particle. They do the same thing with RNA. So they add RNase enzyme, which destroys RNA. They mix the S cells that have been treated with RNase with living R cells, and they still get live S cells again. So something is still being transformed into R cells to allow S cells to appear, and it was not RNA. Finally, in their last experiment, they add DNase to the mixture to get rid of all of the DNA from the S cells. When they mix this with living R cells, then they see finally in this experiment, living S cells never appear. So what this tells us is that DNA was important in order to transform R cells into S cells. And when you don't have it, transformation does not occur. But even though Avery, McCarty, and MacLeod demonstrated that it was probably DNA that entered the living R cells to transform them into S cells, people still didn't really believe this because DNA was a pretty simple molecule compared to something like proteins. So remember, in DNA, we only have 4 different nucleotides, A, T, C, and G, whereas in proteins, there are 20 different amino acids. So people were still doubtful that it was not proteins that were the critical element. So the final experiment that really helped people believe that DNA was the genetic material was performed by Hershey and Chase. So I've outlined the steps here, and I'm going to walk you through the picture in the next slide. So this is Hershey and Chase's blender experiment that they performed on bacteriophage. Bacteriophage are viruses that only infect bacteria. If you ever see the word phage, that means that's referring to a virus. So viruses are pretty simple in structure, especially bacteriophage. They're essentially just proteins. So the proteins often depicted as some kind of hexagon type shape. So this is protein. And then inside they have some kind of genetic material. So to perform this experiment, Hershey and Chase used some important information regarding proteins and DNA. They know that sulfur is found in protein, but not in DNA. So in one batch of the bacteriophage, they labeled the protein coat, the outside shell, which is shown in red here, of these bacteriophage. And in the other batch, the other set of tubes, they labeled only the DNA because they realized that phosphorus is present in DNA, but not present in proteins. So here, the DNA is labeled, and this is radioactive labeling. Then they allowed these phage to infect bacterial cells. And we know that when viruses infect our cells, they inject their genetic material, the nucleic acids. So here I can see that, let me switch to red. The labeling is on the outer protein shell, and the genetic material that's entering the cell is not labeled, right? There's no radioactive labeling entering the cells. Whereas on the top picture, the DNA was labeled, so that labeling is entering the cell. Then they blended the mixture to essentially detach the phage from the bacterial cells, and they get rid of that supernatant, the liquid where the phage shell would be located. And they look at the pellet, the cells within the centrifuge, or excuse me, within the centrifuge tube. When they examined the cells, they saw that there was indeed radioactive labeling in the progeny, so in the viruses that were built within the cell that was infected. So these were labeled. But in the second batch, where the protein shell was originally labeled, there was no labeling. So again, in the tube that contained phage that was labeled with sulfur, the radioactive labeling of sulfur, there was no radioactivity detected inside the cells. But in the tubes that were labeled with the phosphorus, labeling the DNA, there was labeling found in the pellet, in the bacterial cells. This experiment led Hershey and Chase to conclude that it was DNA that's injected into the cells, providing evidence that DNA was, in fact, the genetic material and not proteins. Around the same time Hershey and Chase were performing their experiments, Chargaff was also analyzing DNA, and he found an interesting pattern where the percentage of adenine in a cell was always equal to the percentage of thymines found in the cell, and the percentage of cytosines was always equal to the percentage of guanines. And we know now that this is because A pairs with T and G, guanine, pairs with cytosine. And A pairs with T with 2 hydrogen bonds, whereas guanine and cytosine, or G and C, pair using 3 hydrogen bonds. When we look at this pairing, this can also give rise to Chargaff's other findings. So he found that different species will have equal amounts of purines, purine bases are A and G, and equal amounts of pyrimidines, T and C, but they'll have different ratios of A and T and G and C because of how this binding occurs. Something else I want to point out that's going to be important soon is that each carbon on the pentose sugar is numbered, and they're numbered with number 1. The first carbon is over here, and that's the one attached to the nitrogenous base. Then we have carbon number 2, carbon number 3, carbon number 4, and carbon number 5. The reason that the numbering is important is because we're going to see growing DNA molecules as nucleotides come together. And soon we're going to learn that you can only add nucleotides to a growing molecule of DNA at the 3' end. You cannot add anything to the 5' end because it's already attached to phosphate groups. So you always need an open 3' end of your growing nucleotide or growing nucleotide chain in order to do something like replicate your DNA. So several scientists were involved in the elucidation of DNA structure, and probably the most well-known are Watson and Crick, but Chargaff's rules were important, as well as Maurice Wilkins, who performed different types of X-ray crystallography, and a scientist named Rosalind Franklin that was only more recently recognized for her contributions. She worked in Maurice Wilkins' lab and performed X-ray diffraction experiments to get images like this that really helped Watson and Crick figure out the structure of DNA. So in 1962, Watson, Crick, and Maurice Wilkins won the Nobel Prize for their discovery of DNA, the structure of the DNA molecule. And Rosalind Franklin was unable to get the prize because she had already passed away. So looking at the structure of DNA, I know that DNA is made of repeating nucleotides in a certain sequence that are bound by phosphodiester bonds. And again, this is a review from our macromolecules chapter. Phosphodiester bonds are a type of covalent bond that link nucleotides together to build a chain. For our double-stranded DNA, the two strands run in opposite directions. So this is one strand of DNA, and this is another. One strand always runs from 5' to 3'. The other one will run the opposite direction. And this is known as being anti-parallel. This is required for the bases to pair properly and to generate that really well-known structure of DNA here. We know from before that guanine and cytosine pair. They base pair using 3 hydrogen bonds, and adenine and thymine base pair through 2 hydrogen bonds. If we look at the double helix, the structure of the double helix, the sides, the backbone, which is shown in light blue here, is made of phosphates and sugars. So phosphate and sugar, part of the nucleotide, makes up the backbone, and the rungs of the latter are made from the bases. One of the biggest discoveries in around 1990 was something known as the Sanger method of DNA sequencing, which allowed us to sequence DNA much more rapidly and easily than we'd been able to do before. So let me show you the Sanger method using this animation provided by our textbook, and the link is shown below and in our book as well.
The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all 4 bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Sanger's method is also called the dideoxy chain termination method. So in this method, we use chain terminators, which are called dideoxy nucleotides, or DDNTPs. So these are interesting because they lack a free 3' hydroxyl group end on their 5-carbon sugar. So remember earlier we numbered these sugars. This is carbon number 1, 2, 3, 4, and 5. I said earlier that carbon number 3 has to be open in order for the next nucleotide to attach in the growing DNA chain. If it is not open, then we cannot continue our DNA replication. So if one of these dideoxynucleotides is added to the end of a chain, then no additional nucleotides can be added, and the sequencing, or the replication, the addition of additional nucleotides stops. You cannot continue the process. So in order to sequence a DNA molecule of interest, what we're going to have to do is take that DNA sample and divide it into 4 tubes and add some materials we're going to learn about more in one of the later videos, including primer, the enzyme responsible for replicating DNA polymerase, and some of our nucleotide triphosphates. So we have A, T, G, and C, along with some of these dideoxynucleotide triphosphates, the ones that are chain terminators. So each of these 4 dideoxynucleotides is going to be carried or labeled with a different fluorescent color, a different fluorescent label, so that we can easily identify each of the 4 bases that we look at later on. After the reactions are complete, we use something called electrophoresis to separate the DNA fragments by size, and then a laser scanner reads through the sequence to tell us or report the final DNA sequence of our DNA sample of interest. Here's a picture of what gel electrophoresis of DNA looks like, and we're going to be doing this as one of our last labs in our course. The goal is to separate DNA fragments by size. So it turns out that we start with something known as an agarose gel, and it kind of looks like this. And on one end of the gel, you have little pockets, little rectangular structures, little holes where you can add your DNA. So here we see someone pipetting or adding DNA to the gel. And on one end of the gel, you'll have a negative electrode. It turns out that DNA, I took a little snapshot or a screenshot and I put it there, DNA is also negatively charged. So if you add DNA there, near the negative electrode, and this is DNA, which is negatively charged, and you have a positive electrode down here, and you add an electrical current, then what's going to happen over time is DNA is going to move down the gel toward the positive electrode. So that one's kind of a weird upside down picture. Let's say this is the negative electrode. This is the positive electrode. Up here is where the wells would be, where you load your DNA sample. And then as you apply the current, the DNA will travel down the gel toward the positive electrode. So this gel actually allows smaller fragments to pass through more quickly than larger fragments of DNA. So small, short fragments of DNA will travel down quickly, but the long ones kind of get stuck in the agarose gel, and it takes a longer amount of time for them to travel down. Afterwards, we stain the DNA and view it to compare different fragment sizes of DNA, and we usually run something known as a ladder or a standard. Here looks like there's 2 of them, where you have fragments of known sizes of DNA, so you can look at your fragment of DNA and compare it to a known fragment size. And that takes us to the end of the first video for chapter 14. In the next video, we're going to look at packaging of DNA.